And what they basically demonstrated was that a lot of the slow oscillation is largely driven by layer 5 pyramidal neurons, particularly bursting layer 5 pyramidal neurons. And then this leading to an alternation between an up state, that is a depolarized state when there's a lot of synaptic input and a lot of firing, and then a down state, a hyperpolarized state where there's essentially quiescence. And you can see that as an example in A in this panel from a seminal Stereod paper from 1993, showing this bursting up states with quiescence in between that gives you that slow oscillation rhythm. And by the way, when you look at this, one of the things that often gets a little bit lost is that we tend to think of the slow oscillation, like many oscillations, and as I mentioned at the very beginning, as a sine wave, as a sort of perfectly smooth and continuous event. And it really isn't. It's quite asymmetric. And you can see that asymmetry right in the single cell physiology that underlies it. There's sort of a bursting state that has a different duration and a different onset than does the slower quiescent down state. And so you get the asymmetry from there as well. And this has been not only demonstrated in these sort of animal preparations, but also in vitro preparations and slice preparations in undercut cortex, further suggesting that this is really largely driven by the cortical cells. However, it's also very clear that the thalamus does play a role, that there are very important thalamocortical loops that sculpt the oscillation, that can modify its spatial extent, its frequency, some of the rise times, and there's still ongoing debate as to exactly what and how a given oscillation, a given slow oscillation can be initiated, whether the thalamus can drive that directly or whether the cortex has to drive that. So there's still some open questions here, but this general framework for how this oscillation originates is fairly well worked out at this point in time. But some of the aspects that we originally thought to be the case about slow oscillations, and some of what I mentioned in that first sort of hokey slide with the red curve on it, we think of the slow wave and generally things in the delta range as very widespread and quite synchronized across the cortex. But actually more recent work, and this is a very nice paper from Neer and Tononi back in 2011, using microelectrodes in patients with epilepsy to study the slow oscillations in particular. And they, along now with some other research that's been done, has shown that in fact, these kinds of slow oscillations can be very highly localized. They can be very focal in their origins, and they could be generated by units in a very localized area without necessarily clear generation elsewhere or without synchrony in other areas of cortex. So there's really sort of a shift in or a break in that rule that I first laid down, the first of the rules that I'm going to talk about breaking, that even though it's slow, it doesn't necessarily need to be perfectly widespread and perfectly synchronous. There can be some real focality to it. There are other rhythms in sleep, and we could spend our entire time just talking about not sleep, but even just these elements in sleep. There are things called K complexes, which are not really rhythmic, but have a lot of the same characteristics as the slow wave itself. There are spindles as well, which are a 10 to 15 hertz oscillation that I'll talk about in a few minutes when we get up to that range. There are vertex waves and some other elements that are seen in sleep as well. But the major component that we think about in sleep and in pathology are these slow oscillations in the delta range, in the 1 to 4 hertz range. The theta band is from 4 to 8 hertz. It is seen in drowsiness, but can also be seen in wakefulness. It's also been studied in a number of different preparations, and it could be divided up into a bunch of different subtypes, suggesting perhaps that there are actually multiple thetas with multiple theta mechanisms. One common way of dividing it, or at least thinking about it, is to separate cortical theta from hippocampal theta, again with the idea that there's probably different mechanisms involved. This EEG in the upper right-hand corner is again from a healthy patient showing a fairly pronounced theta activity over a widespread area cortex, but mostly centered in the temporal lobe. The little red box is showing just a cleanest example of that. The other, you see it in other channels on this EEG. I won't yet go through the EEG nomenclature and what indicates what exactly. You just have to trust me for a few minutes at least, that this is a temporal theta for the most part. generated almost certainly from the cortex. On the other hand, the best studied versions of theta really are probably in the hippocampus of mice, where the mechanisms that generate theta still remain debated, and again, probably comes down to multiple forms of theta under different states and different circumstances. But it's thought to be an interplay between some of the intrinsic neuronal properties as well as reciprocal interactions between subregions of the hippocampus, for example, entorhinal cortex communication with the CA1 region, and back again in training the theta activity. It has been implicated quite a bit in sensory processing, spatial navigation, and memory in particular, in both animal models and in humans, but still somewhat of an open debate exactly what role it plays and how it plays that role. And it is also seen in pathology in that excessive theta under certain circumstances could indicate underlying brain injury. So it's not always a functional rhythm as far as we know. Now,
Dr. Cash.
Yes.
Hello. So I'm curious if you can talk at least a bit briefly about how people approach showing the necessity or sufficiency of a specific frequency band in generating these behaviors.
So you jumped to, I think, my penultimate slide,
Okay. All right.
which is right. So I said that it plays some role, and you'll notice lots of hedging words there, and you're going to see a bunch more hedging words coming up. Where I'm uncomfortable saying, theta does X, and that's because while there's a lot of clear-cut associational studies or correlative studies. We do a memory study and we show that when the memory function for a particular stimulus is solid, the theta was high, and when the memory was bad, the theta was low. Or we show that in patients who have trouble with a certain task, their resting theta was not as good, something like that. And then there's a few optogenetic experiments where we induce rhythms of various sorts and enhance memory or enhance a particular processing. Those are the typical ways that it's done, but the majority of work, not all of it, but the majority of work that I'm aware of is really showing a correlation. These things are going up at the same time or going down at the same time. It gets a little bit more nuanced than that, showing some a little bit more statistically interesting relationships, for example, phase locking or destruction of phase relationships or things like that, that can have an effect on the behavior. But again, I think it's one could argue, but it's, I think, mostly correlative, and there's not nearly enough really true functional, sufficient and necessary experiments. And that's because those are really, really hard experiments to do, right? Because it's very, very hard to imagine reducing, destroying, upsetting, offsetting one of these oscillations and not messing up a whole lot of other things at the same time. They're so, intrinsically woven into the activity of neurons in a normal fashion, the interrelationship of neurons, that how do you take them apart without taking apart other things and keep things controlled? So it's a real challenge, and a challenge that labs are getting to, right? They're figuring out tricks to do this kind of thing, and that's the way things are heading. But it's a great question. It's to some extent well, we don't do millions anymore. The billion-dollar question, how do you actually take these things apart and make really strong statements about that? All right, and we'll come back to that at the end of my talk. So now, then there's alpha, and alpha again is sort of the granddaddy of the rhythms. The first one recognized, one of the easiest ones to see. This is an EEG from a person. It actually happens to be from me. Doing a very simple task where I open and close my eyes. And on the left-hand side of this screen over here, you can see that there's some rhythm going on in the background. And then there's this period with the red line here where I open my eyes and that rhythm goes away. And that's the classic alpha rhythm. It's seen at quiet rest with the eyes closed. It's also known as the posterior dominant rhythm. This is really an EEGer's terminology. You won't see that much other place than that, but it's expected to be seen in someone who's healthy and is a good indication of relative health of the cortex, and generally absent or decreased in patients with a whole host of different neuropsychiatric difficulties. Now, the alpha, despite being the oldest and longest studied rhythm of lane, the mechanisms of it are still pretty unclear at this point in time. And again, part of that might be confounded by different forms of alpha, but also because it's just a little bit trickier to look at. There's one set of mechanistic theories that really posit that the pulvinar or LGN is the primary driver of the alpha rhythm, and its projections to the occipital cortex are the main pathways through which the alpha rhythm is initiated. There's a sort of contrasting theory is that it's infragranular layer 5 pyramidal neurons, again, that are mostly responsible for generating the alpha rhythm. And then there's sort of a halfway idea that it's feedback to the upper cortical layers, that is top-down feedback coming from higher area associational areas. And this is a mechanism that we've been exploring. I'm gonna, by the way, I should have mentioned this at the beginning. I'll be showing some data from my lab, not because it is necessarily the best or most important, but it's some that I'm most familiar with, frankly, and lets me illustrate points. But please don't take that to mean that it's definitive in any kind of way. This is work that Mila Halgren did in my group a while ago, looking at laminar microelectrode arrays in patients with intractable epilepsy who are undergoing surgery, and just looking at the spontaneously generated alpha rhythms. And we always saw in these recordings that the majority of the power for the alpha rhythm and the clearest alpha rhythm were in the upper cortical layers near the pial surface, suggestive of feedback information coming to those layers, generating that kind of oscillation. So we think that this kind of alpha that we're looking at is being generated by input from other areas. And in fact, if we look at the how these alpha waves propagate and how alpha itself propagates, it's actually found throughout the brain. Although we think of it as a primarily posterior, that is occipital rhythm, you actually can find it pretty much everywhere in the brain. In fact, there's separate alphas that are prominent in the motor cortex called the mu rhythm. There's a mid-temporal alpha as well, and you can see alpha in the frontal lobe. And it's not really static. It's not that it's just present in one place, but that it propagates. There are sort of waves of alpha activity, and alpha, each individual alpha cycle can propagate. And you see that here. This is a color-coded map of a grid of electrodes that was placed on the cortex of a patient with epilepsy. And again, we recorded during alpha. And the alpha actually starts in the upper right corner of that grid and goes down to the lower left corner. So there's this pattern of propagation of alpha waves that goes from anterior and higher order cortices down to posterior and lower order. And we saw that in multiple patients and also in frontal areas where, again, the propagation is generally from the higher order areas, sort of associative areas, to closer to primary sensory areas or primary areas of cortex. So, one concept then for this alpha, which has been considered to be an important element of top-down cognitive processing, including attentional networks and perception networks, as well as working memory, with this overlying idea that the alpha rhythm is some sort of functional inhibition. It's a gating mechanism. Right. And that's why it's sort of present at quiet rest. As soon as you do anything particularly active, it goes away. And the idea there is when you're at rest and want to be at rest, it's sort of keeping everything tamped down, is keeping that particular set of information relatively constrained. When you start to actually process, now that information is free to flow, and the alpha is no longer needed or no longer wanted to keep that. And that's sort of consistent with this propagation pattern and the supragranular activation. We see that there's sort of top-down control having some sort of a gating effect, or at least modifying inputs into primary sensory areas. And then when the alpha is released, then you can get information through in a different fashion. One possibility is that this takes place during perception. So I show up here the very famous illusion of the old woman or the young woman with a scarf kind of thing. And when you flip back and forth between them, is alpha somehow gating that process, is one of the theories that's out there that we and others are working on to try and understand that. But that's sort of the general mechanistic concepts that are floating around from alpha at this point.